Hi,
Hi, how's it going?
it's going well. Nice to meet you.
Yeah, it's nice to meet you too.
Sorry, I'm looking over the questions right now.
No worries.
Um, so you're the founder of this club, right?
Yeah,
Yeah.
yeah. I am going to San Jose State because they have the microbiology major
Hmm.
that encompasses all the CLS prereqs.
Okay.
I'm trying to get into either their program or San Francisco State's, but yeah, I was surprised that we didn't have a student organization. So I just thought, hey, maybe this is possible. And it would be really, really cool if we could bring in some people who have more experience. And
Oh
that's
yeah, for sure.
glad that we have a chance to talk to people like you.
Oh
So,
yeah. No,
yes.
it's pretty awesome uh that this kind of club existed. And it's like, I guess when I went to school, like I didn't even know about this particular like field that was available. And I think the pandemic has definitely like highlighted it a bit more.
Yeah.
But I do wish that there was more resources for people to know because it is a pretty cool field.
Yeah, yeah. I feel like I hear that from so many people. They have no idea that CLS is a thing, or they think that doctors run the tests.
Oh
So
yeah.
I'm like, no, no.
Yeah. How did you find out about it?
I had first heard about the MLT program,
Hmm.
and then I looked into that, and I pivoted towards CLS because I thought it basically would take the same amount of time.
Hmm.
So yeah, that's pretty much how I found out.
Okay. Yeah, we have a few MLTs that I've taught that are transitioning over into CLS because they wanted to grow a little bit more, and be able to do a lot more of the like complexity testing.
Yeah,
But yeah, I'm unsure if like California has created that bridge program yet or not.
yeah, I've heard about that too. I think, yeah, that would be great because it would just make it a little easier for more CLSs to
Mhm.
not have to go through these really limited programs, you know?
Right.
All right, so I guess we can get started. So I'm recording this, of course, and we do get a lot of views on the recordings, and it
Okay.
totally makes sense to me. Everybody has different schedules, and I think it's good to be able to offer this for people to watch at their convenience. So thanks for letting me record it.
Oh, no problem.
And let's see, let me pull up the questions here. Okay, so I guess I'll just do a little intro. Hi, everyone. Thank you so much for joining us today for our first guest speaker event of the semester. Last semester, we had the opportunity to have a conversation with 4 different CLSs, and this semester, we're continuing our CLS Q&A series, starting with Tiffany, our guest today. And Tiffany has over 11 years of experience as a CLS in the areas of microbiology, virology, and hematology. She obtained her CLS limited license in microbiology at UCLA and focused on virology at UCLA. She expanded her experiences at UCSF, where she completed grad school while working concurrently as a CLS in microbiology. Eventually, she pursued a generalist CLS license at Stanford and pivoted toward hematology. She currently works as a CLS in the microbiology department at Zuckerberg San Francisco General Hospital. Throughout her career, Tiffany has had a passion for education. And in addition to her work as a CLS, she develops CLS curricula, teaches incoming medical residents, postdoctoral fellows, and trains CLS trainees on the bench. So thank you so much for joining us today, Tiffany. We are super excited to have the chance to hear from you and to ask you some questions.
Thanks for
So
inviting me.
yeah, absolutely. Absolutely. I guess our first question would be what initially got you interested in the field of CLS? I know we were just talking about how so many people haven't heard about it before graduating. Was that the case for you?
Yeah, so when I was in college, my counselor at the time kind of asked me during my senior year, like, what do you want to do afterwards? And I knew I wanted to stay in science. I knew that I kind of wanted to pursue healthcare, but I told them I didn't really want to go to medical school. Like, I didn't really want to talk to patients or deal with that like very like face-to-face patient contact. And I told them like I was really good at laboratory, and if there was something that wasn't research related, if there was something like that out there, that I would love to pursue it. And he handed me a pamphlet and he's like, have you heard of clinical lab science? And I told him no. And so I looked into it a little bit more, and I actually got pretty lucky where there is this Biocom Bridge program. It was a grant that was awarded to people in the San Diego County area because I'm originally from Southern California. And it actually fulfilled all of the prerequisites. At the time, hematology was probably the hardest class to find because it was only offered at San Jose State and then one down at SJSU. So after I looked into that and I got into that program, I finished all my prereqs and I did my first cycle of applications. But since I was a microbiome major, I was kind of focused towards microbiology. And that's how I ended up at UCLA in their limited license, thinking that that was what I wanted to do in the future, like just stay very, very focused in microbiology and kind of expand there. And that's how I ended up pursuing my limited license there, starting off with virology. And then I decided, hey, I want to learn a little bit more. And so that's how I actually ended up here in the Bay Area.
Awesome. So you mentioned that you off the bat got your limited license as a specialist in microbiology. Is that the correct way to say it?
So the limited license gives you a more focus. So instead of the 52 weeks that you spend rotating through different sections like chemistry, hematology, microbiology, you delve a little bit further into just microbiology. So it'd be virology, serology, more of the infectious disease side. And I guess it depends on whether or not your hospital offers like mycobacteriology, mycology, and other specialty areas. And so UCLA had developed their limited license program. UCSF also had one where you did 52 weeks of like pure microbiology. And back when I applied, the original like route would be like you would become a generalist and then you pick the department that you really liked and then you sort of stayed in that department. And so at the time I thought I really, really wanted to do microbiology. So I was like, okay, I'll just go straight into microbiology. And it was great at the time because UCLA was developing a lot of the molecular assays. Virology itself was its own department, which was not common for a lot of hospitals at the time because molecular testing was so expensive. So as a new CLS there, I got to see a lot of things in development, such as like MALDI-TOF that we use, and it's like very, very commonly used now. So seeing that kind of like assay development was pretty cool.
So there are some, it sounds like really unique things about, how you really do get to delve into working one particular bench. So are
Mhm.
there only like, are there very few institutions that would allow you to train as a specialist? What's your sense of that in California if someone would be interested?
So I recommend trying to get a generalist license because it offers you more flexibility. You can always take a specialist exam later on after you acquire 3 years of experience in the department that you want. And so, say for example, you start off in chemistry and you really like chemistry, but you wanted to learn a little bit more about micro because you have a generalist license, you can actually jump over to micro. Whereas, like, if you have a limited license, you may not be able to ever do chemistry. So, it's a little bit limiting, but if you really do know what you want to do, and that's where you want to stay, then you can go for it. However, I think a lot of hospitals have transitioned away from the limited license program because having that flexibility to go between departments is a little bit more useful.
I see. Okay, I think that makes sense. So before you became a CLS, did you have any work experience that was relevant to clinical lab science?
Not in a clinical field. I did do research in environmental microbiology as an undergrad, doing a lot of the molecular sequencing and PCR. But in terms of clinical research, no. But clinical experience, I did volunteer at the hospital just to get a sense of what I would be getting into. So being familiar with like what is specimen processing versus like just coming into lab every day and doing like running an assay, like how does this impact our patients?
Awesome. Yeah. So would you recommend that kind of experience for somebody who might be interested in applying to CLS programs, or what in your opinion would be a good experience to go into CLS with?
I think it's always a good thing to be exposed and have a pretty good understanding of what clinical lab science is and how it impacts your patients. So it is quite different from research. There are CLSs that can go into biotech and help with research and development later. But if your focus is more on how do I help the patients and you want to stay more in healthcare, I would recommend, even if you can't get a job as a lab assistant, like maybe reach out to the supervisors and ask them like, hey, like, is it okay if I take a tour of the lab? I could just kind of want to see like how the day-to-day operations is and see if it's like a good fit for you, if it's something that you might be interested in doing in the future. And just kind of like observe what kind of interactions like just the employees have with each other and any of the healthcare employees that come and interact with them as well.
Okay. Yeah, I think that's great advice. Um, so just to go back to what you were saying earlier, since we haven't had the chance to speak to anyone who held a limited license, I'm
Mhm.
kind of curious, like when you did just hold your limited license, what was your day-to-day like with that? And how can you contrast that with what your work is like now as a generalist? What are the major differences?
I think it's still pretty similar because you do work alongside generalists. So, if a generalist decides, for example, they want to work in micro, then they'll still do the same work that you do. The only difference is that a limited license, you'll probably have the advantage of having more in-depth study. So, an example would be in your generalist program, you cover micro for 12 weeks and you rotate through, and they give you kind of like a superficial um, overview of what's happening, maybe let you do a couple of assays during your training. Whereas for me, when I did my limited license, I was on the bench, like I was working up patients and I was consulting with my uh, mentors at the time, how to work things up, what processes that I would be doing just to get me like hit the ground running, basically, after they would hire me post finishing the program.
So now you're working as a generalist, but am I correct in assuming that you're working just in the microbiology and serology departments, or do you get a chance to work all the benches?
I worked mainly in microbiology and serology. So when you get hired, you apply for a specific department because the departments are so large and they cover so many different types of testing. Whatever fits your personality and what you feel like most interested you during your rotations, that's generally how you would pick where you want to kind of start and where you want to, I guess, grow.
Okay, so for a larger hospital, like I assume Zuckerberg SF General is, um, how many CLSs are working in each area? It must be a lot.
Uh, I wouldn't say it's as large as Stanford. I think my current department, I'm trying to think, maybe there's about 15 or 16 of us, and then we each have our own like special bench and we rotate through so that your skills are fresh and relevant. We also do a lot of competency testing to make sure that you're very familiar with what is current and up to date and what is best for the patient.
Okay, that's interesting. I've seen a few other like smaller clinics where there aren't so many people. Maybe there's as many CLSs as you described in your department in the entire lab. And I think that's really interesting to be able to kind of, you're still kind of specializing in a way. Um,
Mhm.
so let's see. So there must have been like kind of a point where you all of a sudden chose to pursue your generalist license. Why was that, and what kind of inspired you to do that?
So when I came to UCSF, I was pursuing my master's in biomedical imaging, which is slightly unrelated to CLS, but it had this infectious disease component. And I really like learning about things that are interdisciplinary because then I can apply it to my work. Like, how do I think outside the box once I have a good foundation for the skills that I have? And as I was working at UCSF, there were some cases where I'd be looking at the patient's history, trying to figure out, was my diagnosis relevant? Is this pathogen the thing that's causing disease in my patient? And I realized there was one incident where I was working up like a urine culture, and I was told like, oh, look at the cell counts. And to me, because that wasn't a component that was part of the limited license in microbiology, the cell counts didn't matter because we were more focused on figuring out which pathogens, which microorganisms, and learning about all the different species. I didn't see the big picture. So to me, I didn't understand until I did my generalist program. Oh, a high like white cell count might be pointing towards infection. If there is an infection, then this high quantity of like bacteria here might be the culprit. And so putting that all together, getting that generalist license actually gave me a more comprehensive view. And I felt that having that under my belt just made me a stronger CLS.
Totally, I can see your logic with that. So it seems like maybe the best course would be if you do have an interest in one particular area, that's always an option, but maybe it
Mhm.
would be best to begin with that more broad knowledge that a generalist license might provide.
Yeah, I
Um,
think so.
yeah, awesome. That's good to hear.
Yeah.
Okay, let's see. I was just curious, because I know, as you said, that you have 11 years of experience working as a CLS, which is awesome. That means you worked during the pandemic.
Mhm.
And we had an opportunity to ask one of our guest speakers last semester about her experience, and it was pretty eye-opening. And I'm wondering what your experience working during the pandemic was like. Where were you working, and what was the effect on you?
So it was interesting because at the time I was pursuing my generalist license. So it was the tail end at the beginning of the pandemic. So as I was finishing up my generalist at Stanford, my program actually got extended because the hospital had to shut down a lot of the medical programs, like including the CLS program. People were just kind of unsure like how long the pandemic would last. Like how long was it until like hospitals are operational? What do we do about this? So my program actually got extended, I think about 3 months or so extra. And so me and my partner who I was with at the time, the two of us did not graduate with our class like when they finished back in like August. So we didn't finish until November, and then we took our licensing board exams in December. But it was a state of like nobody was sure. Sometimes like our trainers would be like, okay, like you have to stay at home and you have to do your study materials there. So a lot of the like on bench lab time, because we had to keep 6 feet apart, was a lot more challenging to finish. And because the state of California does require like certain amount of lab hours that you have to finish in order for you to get signed off. It was a lot of communication, a lot of emails, but we ended up finishing, which was great, graduating. And then because of the short staff, like getting hired was pretty easy.
Mm,
And so we both decided to stay at Stanford because that's where we trained. And
yeah.
so we transitioned over. I wanted to try something new, which is why I went to hematology. And so hematology at Stanford is part of core lab. So core lab means that they did hematology, urinalysis, chemistry, coagulation, all in one area. And so I got to sort of apply what I knew before with my microbiology, like microscope skills. And now I could expand and I could read differentials and look at blood specimens and urine specimens and body fluids and kind of put it all together.
That sounds like it must have been really different at first compared to the work you were doing as a specialist.
Yeah,
Um,
it was different. And then on top of that, the core lab was in charge of the stat testing for COVID. So
oh,
generally, a lot of COVID testing would fall under micros domain because of the molecular testing. But because we also service the emergency department, that actually fell upon us. And since the majority of my team at the time were generalists, they could also run those tests. And so it was kind of chaotic because you were taking on this workload on top of whatever you normally did, which was still pretty high volume.
Yeah. I mean, I imagine that must have been kind of a crazy experience. But I'm really glad that you made it through and graduated and you stayed in the field even. I know a lot of people ended up retiring like at that time, and that kind of makes me wonder, like, is your sense of the field that there is kind of an age discrepancy amongst CLSs? Do you feel like, since you worked at like a lot of different labs too, do you kind of see that there's sort of that older generation and then you have your newer CLSs, or is there kind of a widespread of ages?
I think when I first started, it seemed like there would be like a wave of retirements, but as the field got more popular, I think people are constantly coming into the field, and so there is no real like distinct age groups, I think, and also we sort of all learn to work with each other, and so I think I've been pretty lucky that all my teams have been pretty strong and we've been pretty supportive of each other. So, and I think the advantage of having someone who's more seasoned in the field is that you get access to that kind of knowledge and experience. And so it always felt like, even though if I was the more newer CLS, there was always someone that I could ask, like get a confirmation, a second reading, to sort of like build up that confidence and have those skills. And then now, as I'm more of a seasoned CLS, I can do the same for any incoming CLS as well.
Mhm. That makes sense. So you mentioned that you went through your generalist training as a CLS at Stanford. So
Mhm.
did you complete the didactic portion of the training through San Francisco State or San Jose State?
I completed mine through San Jose State.
Oh,
So it was Mondays would be a virtual didactic program. So we did 4 days of lab on site and then 1 day of lectures. It's a little bit of a different model than the San Francisco State, where they complete all their didactics in the beginning and then they do their laboratory towards the end.
mhm.
But I think I preferred having that like kind of hybrid model
Yeah.
because you basically spend the year long not only training, but it's essentially like because you're doing two different topics at the same time, you're always constantly studying. So by the time your board exam comes around, like you're pretty prepared.
Okay. So at the time that you applied to San Jose State, of course, you were already licensed
Mhm.
in microbiology. So that must have been a huge boost to your application. But do you have any like advice for those of us who aren't coming in with that kind of license, that particular license? What would you recommend that could help build a strong application?
So I'm unsure if having that extra license was a boost because there's such a big shortage. So essentially, from a financial aspect, and this is just my opinion, it would make sense to train someone who was unlicensed because then you have two CLSs in the field rather than one, and then having an additional like person trained. However, I think I was lucky enough that a program did accept me and I was able to grow in my field. For those who don't have the license, I would definitely recommend getting at least exposure to the clinical field. It really does help show when you do apply that you have a very clear grasp and understanding of what you're getting into. A lot of times we do get I do talk to people and they say, oh, like, CLS is not a career for me. This is just a transition point, which I think is a disservice to the field because I think CLSs do work really hard and they are very, very integral in the healthcare system. So, like, I'm very proud to be a CLS. And I think, like, it's a great career to pursue.
Awesome. Um, so in addition to working as a CLS and holding your specialist license and your generalist license, you're also a clinical educator. So how did you first get into that, and where did you first see the opportunity to step into that role, or what inspired you?
So, anytime like you work in a lab, I think that there's always a need for teaching. So I just kind of took the initiative and asked like, hey, can I sort of take charge of the teaching area? And anytime like if someone needed some sort of like clarification, like I would want to not only build my skills as a teacher, but to make sure that whoever was learning from me also got a strong foundation. And I just really like teaching, and I like being very current to whatever methods are being used right now. So I kind of lucked out where I was able to build the like molecular biology section, the lectures for UCSF before I left UCSF. And so that actually kind of tested me to check my knowledge skills to make sure that I was presenting the right kind of material, the most relevant material for the students who are going to use that material. And then going into Stanford, I was able to sort of help like support and supplement their hematology program. So anytime any of the CLS's trainees came in, I would be their point person and make sure that they got the best and the most out of their education, their time that they spent in that department. And the same goes for currently in microbiology, like anytime a pathologist comes in or any sort of visiting resident comes in, if they want to if they want to learn about a certain particular topic. So the last one that I taught was like mycology. If they want to learn about that, I'll sit down with them, prep everything, and then give them kind of a rundown of that particular department and what kind of testing that we do from our end. So it's
You
a
are
great opportunity to sort of like make connections with people. And I think it's a good way if you like to interact with people. So just take the initiative and ask.
awesome. Um, I'm kind of curious, like, what is the connection between pathology and the lab? Are they, if you were teaching pathology residents or something like that,
Mhm.
are we, if we are working as a CLS, are they kind of someone who we might defer to if there's questions about the specimen, or what is the bridge between pathology and lab?
So a lot of the pathologist fellows that come through are training to become like your medical directors. So you'll eventually work side by side with them. In the case you come up with, for example, say you're working on sort of a case on a patient and it's very difficult and you're unsure like what kind of conclusion that needs to be made. Sometimes you have to escalate that particular like specimen or patient and get the medical director involved or your supervisors to sort of kind of collaborate and figure out what is the best like solution for the patient. So a lot of times, if there's something outside of our domain that we don't do beyond our testing, like, and it's going to affect how the patient gets treated, that's a decision that the pathologist will make.
Okay, okay, that makes more sense now. So I heard that you are lecturing right now for the CGMBS program. Is that correct?
No, I am not.
Oh, okay, sorry, that was my misunderstanding. So what kind of curricula have you developed besides the molecular biology curricula that you mentioned?
So currently at SFGH, I last developed the mycology. So currently I'm rotating through mycology because we rotate benches. And so my last lecture was on the section of any sort of fungal infections. And my last resident and I, we talked about The Last of Us, and we talked about how
Okay.
scary fungal infections are and how there was this New York Times article saying that fungal infections might be the next like big pandemic after viral.
Mhm.
So we had this like good conversation. One of the things we talked about was this one species of fungus that we kind of encounter pretty commonly, but it's actually quite dangerous. And she was telling me about like, oh, how her surgery went with one of her patients that got infected with that. And so it was pretty cool to see like, oh, from our end, like we discovered it on our growing on our plates coming from their specimen, but she got to see it like, treating the patient. And then now that that correlation exists, it was pretty cool to see like, oh, this is how we figured out on our end diagnostically, and this is how you see it empirically.
Yeah, definitely. Seeing the all the perspectives that go into treating a patient, I think that would be really cool. Um, so I really am dying to ask you this question. I know I checked out your LinkedIn and You've worked and taught at some of the most notable medical centers in California, from UCLA, UCSF to Stanford, but now you're working at SF General,
Mhm.
which is a county owned medical center. So what is that like, that difference from going from kind of an academic oriented institution to one that is controlled by the county or the state? And what are the like pros that one has, or the cons, or what would you say about your experience there?
So SF General is a little bit interesting because it's still under the UCSF umbrella.
Oh,
So you get a lot of the regulations from county, and then you also have to follow the regulations of the campus side. So you get two sets of training, but being on the public health side, it's faster to access and talk with them regarding any sort of like epidemiological like issues that come up. I guess like in terms of working at different institutions, I would say that each institution was different because their patient population was different.
mm,
So, like LA would have different sets of microorganisms that we probably wouldn't encounter here in the Bay Area, versus like Stanford also had quite a high like cancer related population at the time I was working in hematology. So, if you wanted to study any sort of like cancer related things or see any sort of anomalies, like I lucked out in that they had so many cases because a lot of hospitals do defer to Stanford. So you'll get to see a lot of rare and sometimes very common diseases that you may not be able to see in a rural hospital.
mm.
And so now working at a county hospital, the patient population is different again. So you'll see a lot more patients who don't have insurance, who don't have access to healthcare as readily as people with insurance. But either way, like we still treat our patients as we treat any other person. But I do think that we see a lot more interesting organisms that pop up that I haven't seen before when I worked at my previous jobs.
That's cool. So it seems like most of these facilities too are either tier one or tier two. So you're seeing like emergency situations. So is that the type of training facility that you would recommend to somebody who's maybe just starting out in the field, just to really get a good basis and to see more situations, more interesting specimens? Or what are your thoughts about that? Because in the past, we've heard from some CLSs, they say just get in and get out. You may want
Oh,
a small hospital just to keep your sanity. Other people have said, uh, like that was what I was really interested in, kind of like the more intense environment of hospitals that are tier one or two. So what are your thoughts about that? And what do you think is best from a training perspective?
I think when I first started out, my goal was to try to learn as much as I could within a short amount of time, because I felt that once you started going into the routine of things, sometimes due to scheduling or staffing, it might take longer for you to get trained. But anytime there was some sort of opportunity to grow and to learn more, like I jumped on it because I felt that I wanted to be really good at what I did. And so for me, I really enjoy learning very deeply into my subjects. So since microbiology was my starting point, I wanted to learn really well, like not only bacteriology, but virology and all the little like subsections because I was a limited license. And I felt that having that really good foundation at first is great because once you have that foundation, now it enables you to kind of think outside the box. Like, how do I improve the field if that's the direction that you want to go? Like, if you have better ideas on improvement, like, how do I make my team stronger? And so I think um, if you are unsure of which department you want to go to, I would start as a generalist, kind of in a core lab, and see like what the best fit for you is. And it's highly dependent on each person. So if you're kind of more of a chill, relaxed kind of person and you don't really want to, you're not like I guess familiar or like easy with like the grind or high volume, then maybe a smaller hospital is a better fit. Maybe biotech is a better fit. If you want to see as many things as you can, maybe a high volume hospital like with the academic like side might be better. So the advantage of working for a school related institution is like you not only do your CLS work, you get to see like assay development. You get to interact with other people. There's like a lot of different things that you get to do instead of just your day-to-day work. So if
Mhm.
that's something that is I guess like appealing to you, then I would pursue sort of in that direction. But it's highly dependent on the person.
Yeah, yeah, that totally makes sense. And I think that's kind of one of the reasons why it's so great to hear from CLSs like you, because it really is eye-opening to see all the different directions that you can go into in this field.
Mhm.
I think when you first hear about it, you imagine yourself working in this one environment, in this one position, but there's so many different ways that you can take this career. So kind of speaking about that, how do you see that there's chance for mobility in this career, upward mobility, and if so, what kind of training do you think that would take? And I guess just anything else along those lines that you'd like to add.
Uh, yeah, I think there's a lot of different opportunities, like as you gain experiences. My current focus is to sort of specialize and get towards like a specialty, and be more like a point person for younger CLSs that are coming in. But that's just my passion because I want to be able to train new people and make sure that they feel really confident with the skill set that they have, no matter where they go. And so, yeah, I feel that like I think there's a lot of opportunity in different I haven't explored biotech, so I'm unsure, but I have friends who have gone to biotech and they really enjoy it. If you're more into like the research and development side, like that is something that you can apply your CLS knowledge to help develop the new assays that might get used in the hospitals later. And
Mhm.
so I think there's a lot of opportunity there. And if you want to do more of the leadership, like laboratory management, I would suggest like maybe getting more into like, I guess, like working with your supervisors later to sort of see the more administrative side, if that is something that you're interested in.
Mhm. So if somebody would be interested in kind of pursuing a path similar to yours that mixes in some clinical education with just working as a CLS, what are your recommendations for how to pursue that?
You can, I think just being a bench tech is good. And then as you gain more, you can ask and take more initiative to be like, oh, how do I improve this process? Because a lot of times, like, because the environment is so busy, it's good to take initiative and just kind of do things on your own and make sure to work with the people that you're working with. And you'll get a lot of ideas with your team. And together you'll kind of grow together and kind of build this like product that you really really enjoy, and then you'll be able to share that with incoming visitors and CLSs. So it sounds like you're really interested in education as well.
I might be, yeah, and definitely it's a possibility. But I really like hearing about your experience with it and um, just helping to prep CLSs to be strong in whatever job they end up taking. I think that's really cool. So I'm curious, so now that you're working at SF General, in the microbiology and serology department, what is like a regular shift like for you? Like what sort of things are you doing throughout your shift?
So I'll come in the morning and then you check your schedule to see like which bench you're assigned, based on where you're trained. SF General does like a monthly rotation. And then within the month, like say, for example, I was in mycology. So that would require me to do the day-to-day, like checking my fungal cultures, working up anything that I see that is an anomaly. So if it's depending on the site, like mold is generally not supposed to grow in your body. So I'll work those up, I'll identify those, I'll alert the physician. And if there's any sort of like treatment needed, say like drug testing, like the physician will call me back and we'll talk about it. And then either we'll send it out or we'll continue testing. And so that's like the general day to day. If we have visitors, my supervisor will call me and say, hey, on this day, we're going to get like a pathology resident. And since they're going to be rotating through your section, like, can you prepare like a lecture for them? And so then I will either grow everything out or find or build like a PowerPoint for them to sort of encompass whatever I'm working on at the moment. And then for me, I do one other thing where I reach out to that particular pathologist and I'll ask them like what their interest is so that I can sort of curate how I'm going to teach for that particular pathologist. Because the curriculum for pathology is they'll rotate very similarly like we do in CLS. So they'll go through chemistry and hematology, and then they'll finally decide where specifically in pathology they want to end up. And so sometimes you might get a pathology who may not be interested in micro and more interested in chemistry. Then for me, I can sort of structure my lecture to give a more superficial overview. But in the case like a pathologist is like, oh, like I really, really love micro, like I've never seen this kind of thing, like can you show me more? Then I'll go a little bit more in depth and show them a little bit more things that we encounter that are unusual.
So that kind of makes me, it brings to mind like the fact that you have your limited license in addition to your generalist license. Are you able to leverage that for your salary or your pay, or you don't have to share that if you don't want
Oh,
to. I'm curious from that
to my
standpoint.
knowledge, currently, no. It is something that we're working on.
Mhm.
The specialist license is something that I personally wanted to pursue,
Mhm.
just so that for me, I liked to know that how well I was grasping the material and if there's anything new, because every year the ASCP will sort of reevaluate, and then depending on which cohort you're in, they'll release the guidelines of what you need to learn for your test. And so I'm constantly monitoring that so that when I do teach my students, they are currently up to date. And if we don't have whatever is covered on there, like I'll do additional research so that they are at least covered for that portion.
Awesome. So you've worked at, how many labs total have you worked at now in your career?
I think SFGH might be my fourth lab.
Fourth lab. Okay, so
Yeah.
that's a lot of labs. Um,
Yeah.
which was your favorite, and in your opinion, what makes a given lab a good place to work?
Hmm. I don't know if I had a favorite because each of them had its merits. I
Mhm.
think the overall like theme that I can think of is they all had very interesting like things to learn because the patient populations were so different. I felt that there's no one individual experience that said that this lab was better than this lab.
Mhm.
Um, Thankfully, I was pretty fortunate that all my teams were very, very good. I thought that a lot of the seasoned CLSs that I worked with had a lot of great knowledge and experience to pass down. And so for me, it made the transitions very easy to like get on board with that team. I think it's a little bit tougher, say, for example, if you're working at a startup and you're the lead, like it's hard to sort of figure out what to do, especially if it's like a brand new environment for you.
Right. That makes sense. Um, so in all the places that you worked, were you required to work weekends, holidays, that kind of thing, or were there unions that you could join? Um, or I guess, do they all offer pretty equal benefits and that sort of thing?
So Stanford was the only place that did not have a union,
Mhm.
because the UCs are considered a government entity. They actually had their own union.
Oh,
Um, so I'm trying to think. Benefit wise, they were pretty similar. The advantage of working for like a UC or a government entity is that there is a pension. So
mhm.
if that is something that you are looking to think about in the long term versus like investing in a 401k, that is something to think about. For the UCs, it takes 5 years to vest into your pension to start it off.
Mhm.
However, I feel like overall the benefits are pretty similar. I do have to work weekends. The rotating schedule depends on how large your staff is, what the needs are of the lab. and whether or not you're short-staffed or not. So if you are short-staffed and your particular department has a lot more stat testing, then more likely you're going to have to do more coverage. But overall, I feel like the system is fairly fair in the teams that I've worked on, where you are rotating in like equal parts.
Okay. And we've kind of heard some mixed information in our classes about like whether or not being a CLS can really be whether or not it provides you with a livable wage, especially in the Bay Area where it's incredibly expensive to rent, to own a home, just to eat. So in your experience, is this career something that provides a livable wage?
Um, I think I think for now, most hospitals that I've worked with are, especially the ones that are unionized, are trying to get a lot of the wages up to market rate.
Mhm.
And so I think that is one of the benefits of having a union because then they can fight for that wage discrepancy. Whereas if you're working in a more private institution, the company itself has more power over how to adjust your salary. Um, I think I haven't worked in any place that wasn't paid hourly. Because I think if you are paid salary, they might have to accrue overtime that may not get paid.
Right.
So, yeah, I would recommend working for an hourly wage.
Yeah, that sounds like the right call. So now that we've heard about kind of the path of your career and a little bit of advice you have, just looking back, what would you say so far in your 11 years of experience, what has been the most challenging for you about being a CLS?
Challenging. I guess sometimes the workload is very high, but that just happens to come with the institution that I worked at.
Mhm.
So sometimes it is hard to sort of, especially things as instrumentation starts becoming more frequent in all the departments. A lot of the instruments take a lot more maintenance. Though the results may come out faster, I think maintenance, and especially when you have troubleshooting errors, that does cause a lot of delays. And that is probably the more stressful thing for me, is spending time to fix the thing and not being able to get my results to my patients on time. And so when it becomes really hectic and also having the more patient facing side, not being able to see what you do, it is more challenging to kind of communicate that with them because there are misconceptions for CLS where they just kind of send off the specimen and magically like a result appears. They don't understand that it takes a lot of time and a lot of thinking, interpretation to actually get you that result. Which is why I appreciate working in a hospital where like the doctors can come down and like chat with you and they do sort of understand like, oh, okay, like this is the work environment that you're in. And they're pretty good about like calling you as needed rather than like being off campus, for example.
Right, like in a reference lab or something like
Mhm.
that. That makes sense. So in contrast, what to you has been like the most interesting thing in your time as a CLS so far? Or are there any like moments that really stand out to you where you're like, oh, that is so awesome or so interesting, or I just love that?
I'm curious to see where technology comes into play. So, like one of the instruments that I use in hematology, the Sysmex, basically classifies all the blood cells based on like the picture. So, it'll take a snapshot of your slide and then it'll sort out all the like different white blood cells. And to me, that's pretty interesting because when I was in grad school, like we talked about biomedical imaging and how to apply that with like I guess the buzzword machine learning and AI, and that is one component where you can kind of train your computer to like sort things out and like kind of classify things to help speed up your work. So I think as new technologies are being introduced and implemented, it is pretty exciting to see like how much more efficient we can be and how much more accurate we can be and spending more time in our skills helping our patients rather than like something like oh machine maintenance or something like that.
Yeah, I agree. I think that's really going to be something interesting to keep an eye on in the future. And I'm wondering, does the lab that you work in now have any equipment that is able to automatically identify plated specimens? I've heard there's some kind of machines that do that, but I've also heard that they require a lot of maintenance.
So we have the MALDI-TOF for bacteria where you can kind of spot it onto this plate and it uses mass spec, and based on the spectrum of that against the algorithm, it can give you with pretty good accuracy what it is. And that actually speeds up the identification instead of doing the traditional way, which is like setting up all the biochemicals, waiting for it to grow. And now some laboratories have gone even more advanced, and you can go straight from specimen to PCR. And so now that you have that genetic material available and you can multiply that and figure out like what it is, like now you can get the results even faster. And one other step that you can take is auto verification. So say you set up the PCR, instead of you having to manually enter all the results, now it'll go straight into the patient's medical records. So then now the doctors can see it right away as it finishes.
That's awesome. Yeah, I think there's definitely a lot of room to improve efficiency in the lab for sure. So you have mentioned your biggest challenges, some stuff that you found the most interesting. And what do you feel like so far has been like the most important thing that your time as a CLS has taught you, or what's the most important thing you've learned?
Mm, I learned that communication is key. So to be able to not only communicate your results, but to work well with your coworkers, because there's a lot of stuff going on in lab. It's very chaotic. Especially, I heard, for example, like blood banking. If you have a trauma and you're trying to, everyone drops everything to try to match the blood. Like that's a lot of chaos, especially if it's a huge trauma. More specifically in like microbiology, if a stat testing comes in, then you need to be able to address it while doing your other work, but you need to also communicate to people like when you need help. So if there's something that's more pressing, working well with your coworkers to ask them like, hey, can you take over this? And then I'll get back to it after I finish this stat. And just so that there is a continuous flow of work, and having that good line of communication, not only with your coworkers, with the rest of your team, your supervisors, is really, really important.
Awesome, that's good to know. So what advice do you have for us as people who are interested in applying to CLS programs or interested in this field? What advice do you have for future applicants in how we can prepare, or what we should be prepared for mentally, or anything like that?
Uh, so I guess there's the four basic state requirements, which is your medical micro, your immunology, your analytical chemistry, and then your hematology. Those are pretty much the core courses that you're going to have to redo when you go through the program, but you're going to study it more in depth. And also, I would recommend maybe taking some elective classes, just so you can broaden like your repertoire of like what you may encounter, because it's going to all help you and add up in the future. And then definitely get some clinical experience if you can. um, Ideally we had a lot of lab assistants who got their clinical lab experience that way, and it actually did help them a lot as they applied for their programs. And if you can't get the lab assistant kind of experience, at least like see what a clinical lab day to day looks like, whether it's volunteering or just kind of doing a lab tour and getting that understanding. Because oftentimes you may think that you might like it and you might want something more, or it might not be what you imagined. And once you get into it, maybe like it's not a good fit for you. So just be really sure that this is what you want to pursue, and just get a good idea and like keep an open mind.
Okay, I think that's really good advice for um, potential future applicants. So for people who have just become licensed and they're just about to get their first job, what advice do you have for them? What do you think would be the best path to pursue directly after graduating?
So once you get your first job, like, um, be very open to learning. Try to learn a lot of things and learn them really well. And it'll help you build your confidence as you start out as your CLS. And don't be afraid to ask more seasoned CLS, like, about their experiences. about like how they would approach a certain specimen or a patient, because they may have seen something that might have worked better, and to really follow your procedures because they are written in a certain way such that you get accurate testing. So be very meticulous in your work, be very open-minded, and be ready to learn.
And then I know we had kind of talked about this a little bit earlier, but do you feel like this profession offers good job stability and got a good work-life balance?
Job stability, I think, yeah, I think it has a very good outlook for the future. There's definitely like a huge need for CLS. And I think, unfortunately, there aren't as many training programs um, to fill all the need for the CLS. So I think it'll definitely be great opportunities in the future once you complete your program. For job stability, besides that work-life balance, I think you just have to work with your team. So like the downside of working in a hospital is that your laboratory is going to run 24/7. And so you need to be pretty flexible with how you operate. If you want a more stable schedule where it's like, I only want to work Monday through Friday, like maybe the hospital is not the best environment for you. However, I do recommend personally to start off at a hospital because you'll see a lot more and you'll be able to apply those things, even if you do deviate away from the hospital in the future. At least you'll have that like solid foundation.
Yeah, that totally makes sense. So we were talking about kind of AI and technology in the future. How do you see the field changing? Is that the biggest way that you see this field changing in the coming decades, or what are your thoughts on the future of CLS?
I do see a lot of automation coming, and I do see a lot of fear of automation coming. So there is a little bit of resistant, like there is that fear of, oh my gosh, like the robots are going to take over my job. But for me, I see it in a different way where it's like, okay, the robots will be able to do things that will free up my time so that I can focus more on my patients. So
Yeah.
to be able to get rid of the like more menial day-to-day things so that I can actually use my skills and my knowledge to help the patients and actually like improve the field. So I think it's a good thing.
I like that hopeful message. That's good. So is there any other info that I kind of missed that you would like to share? Anything that you feel is important for future CLSs to know? Anything along those lines before we
Unsure. Do you have any questions for me? Any burning questions?
go? I mean, I could go on, but I want to be respectful of your time. I guess if there's anyone else here who wants to ask a question, we have this awesome opportunity to ask Tiffany literally anything. Well,
Yeah, and feel free to reach out. I'm pretty good at like responding back.
awesome. Okay. I can definitely share your email so that if people have questions when they do view this, they know they can reach out to you. And that's really awesome. Thank you so much for offering that.
Oh, no problem. Just let me know that this is where you we talked in the case there's like a spam email or something like that and I don't accidentally like miss it.
That's a good point. Yeah. Okay, I'll share that. Um, yeah, if anybody here has any questions, you can just unmute yourself and ask. Otherwise, I will share Tiffany's email. Yeah, this is such a good opportunity. I'm so glad we had a chance to have a conversation. I was really sad that I didn't have the chance to talk to you earlier. So thank you so much for waiting until September, because I know we had talked back in April, I think it was. So this was such a good start to our club guest speaker Q&A. So
Oh, thanks for inviting me.
yeah, thank you. I'm really stoked that we had this opportunity. You gave us so much info, and it's really just like demystifying the field to so many of us who have just heard maybe that there is a program at San Jose State, but we don't really know, what it would look like on the other side. And this helps to just really like round out our perspective of the field and just I think it's such an important missing part of like potential applicants, like we just don't get a chance to hear from pros like you. So yeah, thank you so much.
But one thing that I would recommend is if you are interested, you could probably look at the ASCP website.
Okay.
They do have, if you are starting out as a CLS, they do have scholarships and they do a lot of growth opportunities. So if that's something that you're interested in, I think you mentioned that you were a student still. I think they might have some like scholarships for students as well who are interested in the field. So check that out. I don't know if they update it as often, but I do know they exist because there is and especially like if you end up specializing in a certain area, they also have professional societies as well. So for example, microbiology has like the American Society of Microbiology, and they hold yearly conferences. So one of the things that I did was they did have a grant for young professionals, so early career CLSs. And so when I got selected for UCSF, the good thing is that they will give you not only like a financial like stipend to spend, but they actually partner you up with a mentor who's very seasoned in the field and probably has made significant contributions in the field. So it's and you go around with them during the conference. So that is a good opportunity if you want to sort of grow in your field in a particular section. And so each of the different departments should have their own like societies.
Wow, that sounds really cool. I had no idea that even existed. I'm definitely going to share that with everyone so that we know about that opportunity. So you're saying through ASCP, there's grant opportunities or scholarships for undergrads potentially?
Mhm. And
And
also, I think incoming CLS students.
okay, cool. Awesome.
And then since you mentioned you were in microbiology, Northern California has Northern California ASM as well, which is the northern half of California. They do meetings every once in a while. So if you are interested in just seeing what's happening out there, they do have student discounts for that as well.
Are those in-person events at this?
Mhm.
Awesome.
Yeah, so it's like 1 or 2 days. The one in Southern California is usually held in San Diego, and they'll bring in speakers, and they sort of give talks about what's relevant and what's current in that particular field of the clinical area. And then once you are licensed, you are required to renew your license every 2 to 3 years, depending on who is your licensing agency. And you have to submit CEs, which are continuing education units. And to get those, you either do online classes or you can attend these kinds of functions where they will give you contact hours.
Cool. I had no idea that the conferences would count for that. That sounds like definitely a more interesting way to get CE credits.
And you get to meet a lot of people who are in the field as well. So it's fun to see new faces and familiar faces.
Cool. Are you planning on going to any of those this year or next?
I think ASM just passed. That one actually jumps around. So I think the most recent one was in Texas.
Mhm.
I think the next one, I'm unsure where it's being held, but I do recommend going just to see like what's out there. Like you get to see all the vendors, you get a lot of swag, and you get to see a lot of people who are interested in that particular field.
Do they present like new research in the
Mhm.
field or
Yeah.
the speakers? What do they focus on?
So there's like multiple speakers going on at the same time. So like if you're an undergrad, you can present, they have poster sessions for undergrads and graduate students that are doing their research. There's also talks going out throughout the day. So for example, if there's a talk about like antimicrobial susceptibility, they'll talk about what drugs are currently being used, what's no longer appropriate for usage, and just kind of like an update for that. They'll also talk about maybe like specific organisms that they found or things that are happening across the world. And so, depending on the speaker and the topic, like you can just pick and choose which ones you want to attend and just kind of sit in on them and just kind of absorb everything.
I really like the idea of these conferences, especially with what you were saying about, working to just be as knowledgeable as possible in what you're interested in as a CLS, so that you can provide really good care. I think that sounds like a good way to achieve that and just stay up to date for sure. That's cool. So there's every year it's in a different city nationwide.
Yeah.
Okay.
So I think ASAP also holds their own. I haven't been to that one. I've only gone to the one for ASM, because that was relevant in the department that I was working in at the time.
Mhm.
But I do know, I think chemistry also holds one. There's one for blood banking and hematology as well.
Nice. That's cool. That would be awesome to attend. And maybe one year I will see you there.
Oh yeah, for sure.
Cool. Well, thank you so, so much for taking the time just to answer all of our questions.
Oh
And
yeah.
it's really, really good insight. And I'll share your info with club members if that's okay with
Oh,
you.
for sure.
And I might follow up with a few questions if that's okay with you as
Oh,
well.
absolutely.
Cool. Again, thank you so much, and I'll let you go so as not to run over time too much. But it was awesome to meet you, and yeah, I hope we get to talk together soon.
Okay, thank you so much.
Yeah, thanks.
Bye.
Bye.